Dr. Kenyon, what first interested you in origin of life research?
Well, I've had a long-standing interest in the life sciences, and it goes back to high school days. When I got to college as an undergraduate at the University of Chicago, I was studying physics at the time, but the Darwin Centennial celebration took place there in 1959, and I had an opportunity to hear some great people speak on the subject of origins, and really piqued my interest and kind of had an impact on me for deciding what to do in graduate school, going on to Stanford and working with some people who were working on the origin of life problems.
What was your viewpoint on the origin of life when you wrote Biochemical Predestination?
Well, at that time I was working as a postdoctoral fellow in Melvin Calvin's laboratory at UC Berkeley in an atmosphere where there was a great deal of work and discussion on the origin of life problem from the perspective of chemical evolution theory. And I had that view that life did in fact arise on the planet by a chemical evolutionary process. I was pretty convinced that in fact was the case.
How have your views on the origin of life changed since you wrote Biochemical Predestination?
Well, in the years following publication of Biochemical Predestination, I had the opportunity to teach in the area of origin of life and in the area of Darwinian evolution here at San Francisco State. And after about 10 years of this teaching activity, I began to have some doubts about whether or not life could have arisen by natural means, by chemical evolution, and I had some doubts, growing doubts, about whether or not there was a Darwinian process to generate the major forms of life. And so it took a while to reorient my thinking, but I did eventually change my views in both those areas. Yeah.
Do many of your colleagues support your new position? If not, why not?
Actually, very few have been supportive of my new views on origin of life and the development of the species. I think a rather larger number of my faculty colleagues, however, are willing to allow me to express these new views in class. But the majority, though, are unhappy on both counts, I would say. And as to why they hold this view, I don't think there's a simple answer to this. Some of the possibilities are that the Darwinian way of thinking is such a deeply ingrained habit of thought. It's been prevalent for such a long time, and it has so many implications for the whole of the biological sciences. Another aspect tied to that, of course, is how graduate students are socialized as they go through their program and hear only the Darwinian story of origins. And I think my colleagues in general, those who are opposed to this, are very reluctant to take time to examine this issue, and maybe they realize the large reorientation of thought that would be required if they were to change their views. So.
What are the general presuppositions that scientists make who study the origin of life?
Well, I think there are two most general kinds of presuppositions that people can make. One is that life, in fact, did arise naturalistically on the primitive Earth by some kind of chemical evolutionary process. The second presupposition would be that life may or may not have arisen by a naturalistic chemical evolutionary process. Now, if you have the first presupposition, then the goal of your research is to work out plausible pathways of chemical development to go to the biopolymers, then to the protocells, and what would be likely pathways that you could demonstrate in the laboratory by simulation experiments. If you have the second presupposition, you're still going to be doing experiments, but you're going to be more open, I think, to the possibility that the data, as they come in from those studies, may actually be suggesting a different explanation of origins altogether.
What is the Oparin-Haldane hypothesis, and what role does it play in current research and teaching on the origin of life?
Well, the Oparin-Haldane hypothesis is the most general theoretical framework within which experiments and research on the origin of life are conducted still today. Oparin proposed his chemical evolutionary views for the first time back in the 1920s, and Haldane right at the end of the decade of the 20s. And essentially the hypothesis says that on the primitive Earth, before any life was present, you had carbon compounds in very simple form in the atmosphere, in the form of methane, for example, and other simple gaseous substances, and that these would gradually increase in complexity under the influence of energy provided in the natural environment, solar ultraviolet flux, for example, cosmic radiation, heat energy. And then you would get the transfer of these molecules, somewhat more complicated molecules, down into the oceans, amino acids and sugars, for example, and then these would link up later on into polymers, polysaccharides, proteins, and then you could have nucleic acids forming. Then you get protocells from aggregation of these materials. They would compete by a kind of proto-natural selection process, a proto-Darwinian process, and the one that would win that competition for dwindling nutrition in the oceans would be the first to arrive at the state of a full living cell. So it was a process of very gradual complexification of carbon compounds, taking maybe hundreds of millions of years.
What are the major underlying assumptions of the Oparin hypothesis of chemical evolution?
Well, there are a number of them. The first that I could think of was that Oparin thought that the ancient atmosphere was very different from our present air. It was a reduced atmosphere, and it did not have molecular oxygen in it at all. Typical gases were methane, ammonia, water vapor, and molecular hydrogen. Another assumption would be that the molecules that were formed in the atmosphere under the influence of these energy sources were somehow protected from destruction by those same energy sources, so that you could really have an accumulation of life-building substances, amino acids and sugars and purines and pyrimidines, could actually survive the rigors of the ancient environment. Once they got into the ocean, if they ever did, another assumption of the hypothesis would be that just the right what we call biomonomers, amino acids, for example, or sugars, would react with the same kind of biomonomer to give you the kind of polymer that is so characteristic of life, namely a polypeptide, all amino acids. In other words, no other substance would get in there and interfere with that clean production of polypeptides.
Are there any other important assumptions in origin of life theories?
Yes, there are other basic assumptions of the Oparin hypothesis. One of them, additional one, is that that just the right optical isomers of, for example, amino acids reacted with each other like all L-amino acids, forming the first polypeptides. Since we find exclusive currents of the L or left-handed form of amino acids in protein today, Oparin had to assume that there was some sort of process by which the L form would be separated out from the D form, which is equally likely to have been formed. We also have to assume, if we believe this is the kind of process that generated the first life on the planet, that the polymers that were forming in the oceans could have acquired genetic information. And this is a key point: specified complexity, genetic information in these polymers. They have to, the subunits have to line up in specific order in order for biological function to be possible. And yet we know of no chemical means for the alignment of these subunits in the absence of pre-existing genes. And of course, when we're going toward the origin of life, we certainly don't have any genes in the picture. So the question is, how can these substances align themselves? And so it's a major assumption of the hypothesis. Another one is that as you get close to forming an actual cell, that all of the many, many different components you need for a cell to live, all the different enzymes and the nucleic acids and the other substances and the metabolic reaction sequences could all have originated simultaneously and in the same microscopic region of space, a bacterial volume. And the probabilities against that happening spontaneously would appear to me, do appear to me to be overwhelmingly negative.
How well are these assumptions supported by currently available scientific data?
Well, let's start with the assumption of the reduced or reducing atmosphere of the primitive Earth. I think that if we look at current geological data referring to the oxygen content of ancient surface minerals, we cannot decide the issue of whether the ancient atmosphere was reduced or not, but the evidence is consistent with the possibility that there may have been some oxygen present in the old atmosphere. Another reason for believing in the strong possibility of molecular oxygen having been present from the earliest times in the atmosphere is the fact that even today in the high atmosphere, water vapor is being photodissociated by the solar ultraviolet radiation, and the result is molecular oxygen and molecular hydrogen. Hydrogen is very light, could have escaped into interplanetary space, leaving that oxygen there, I think, from the very earliest times. So if oxygen had been present, even to the extent of only 1% its level, or 1/10% of its level in the current atmosphere, I don't think you could have had any chemical evolutionary development at all, because oxygen destroys organic compounds very effectively. So you could say that chemical evolution, according to the Oparin hypothesis, would have been extinguished right at its source, right at the earliest stages of its movement.
What is your evaluation of the Miller type of simulation experiment?
Well, I think Stanley Miller's pioneering simulation studies, at the time they were done, did provide some very valuable insights into the possibility that a chemical evolutionary development might have occurred on the primitive Earth. He did discover in his apparatus the synthesis of several protein-forming amino acids and a few other substances, the type that occur in cells. I think in subsequent years, though, as time has gone on, we've looked at these experiments in the light of new scientific data, that they appear to be less and less relevant to the question of origin of first life. There is the issue of the composition of the atmosphere, and the crucial question of molecular oxygen, which is routinely excluded from all of the Miller experiments, his original ones and all of the follow-up studies that have been done since that time by others. And if oxygen is present in the apparatus, you're not going to see any synthesis of amino acids. You better also not include molecular hydrogen, as Miller did in the first study, because then you'd have a great explosion in the laboratory. So I think that we do have reason now for doubting that these studies tell us much about the possible origin of life. I think most of the chemical reactions that go on in this apparatus are away from life. The substance that appears in the apparatus is a kind of, that coats the whole inside of the apparatus, kind of this amber, intractable macromolecular material that is non-biological, doesn't occur in any organism, but represents 85% or so of the converted carbon that you start with as methane. So the trend in these experiments is really not toward biochemistry, but in my view is away from biochemistry.
Is it possible that interfering cross-reactions might prevent life from arising naturalistically?
I think that's very likely. The most prominent interfering cross-reaction I can think of at the moment is the so-called Maillard reaction. Now, this is a reaction that happens between sugars on the one hand, or aldehydes, and then on the other hand, amino acids or amines. And these are two classes of substance that would be very important in any chemical evolutionary development toward life, and their tendency is powerfully to react with one another rather than for the amino acids to be able to react with themselves on the way to protein, or sugars to react with themselves on the way to polysaccharide. The cross reaction to form an insoluble polymeric material called melanoidin is far and away the more probable reaction, and this is very well documented in the literature. This reaction, the Maillard reaction, has been known for many years, and in fact is the reaction that is involved in the browning of foods when you cook and do baking and so on, and also the brown color in various beverages is a result of the cross reaction of amino acids and sugars. I believe that kind of thing happens in the Miller simulation studies, but what it does is it takes you away from the development toward the first toward living matter, and kind of locks up the organic matter in this inert form.
Stanley Miller's pioneering work in the origin of life assumed a reducing atmosphere of methane, ammonia, water vapor, and carbon dioxide. Is there sufficient empirical support for this assumption?
I think there's not sufficient empirical support for this assumption. I think, if anything, the balance of evidence suggests that it is wrong, that it seems more likely to me, from the geological evidence that we have about very ancient sedimentary rocks and their oxygen content, and about the photodissociation of water vapor in the upper atmosphere, that the early atmosphere would be far more oxidized than the early Miller experiments assumed, but even more oxidized than the current Miller experiments. He's using carbon dioxide instead of methane now, and he's using carbon monoxide and molecular nitrogen instead of ammonia now. But I think that the crucial question is still the absence of molecular oxygen. They're still routinely keeping that material out, and the reason seems to be not an empirical reason, but a methodological necessity, if we could put it that way. In other words, if the argument, as I understand it, goes something like this: if molecular oxygen had been present, then chemical evolution could not have happened. Therefore, molecular oxygen must have been absent, because we know that chemical evolution happened. But I think it's important to note that we do not have independent or convincing independent corroboration of that absence of oxygen.
Do Sidney Fox's proteinoid microsystems represent a plausible laboratory model of the protocell stage of chemical evolution?
Well, Sidney Fox's proteinoid microsystems are indeed very interesting experimental model. I'm impressed by the fact that you can go in one continuous sequence from amino acids all the way to microscopic units of bacterial dimension. When you look at these microsystems under the microscope, they indeed resemble, the light microscope level, spherical bacteria in size. But I think the resemblance stops there. I believe that I know that if you look at this into the chemical composition of these microsystems, you find very little that reminds you of the chemical complexity of cells. There is generally no protein present. Proteinoid is present, but that is not the same as genuine protein. It does have amino acids in it, but there are many chemical differences between the proteinoids and protein. There are no nucleic acids in these systems, no polysaccharides, no lipids. There is no metabolism going on. They're chemically inert. There's no genetic information whatsoever, in my view, in these microsystems. They are not self-replicating units. They have no energy capture or processing capability. So when you run down that list of negative features, you come away pretty much with the view that these proteinoid microsystems probably didn't have anything to do with the origin of life.
How large a gap is there between the most complex protocell model and the simplest living cell?
Well, I think this gap is an enormous gap. You have the great difference between the most complicated protocell and the simplest living cell, which would be something like a mycoplasma type of organism. You just don't have any of the ultrastructural detail in the protocell models. You don't have any ribosomes, you don't have any nucleoprotein particles of any kind, you don't have any nucleic acid in there. You just have a simple little microscopic inclusion in these protocells with virtually no lifelike properties. So I see the gap between the best protocells that have been produced in the laboratory and the simplest authentic living cells we have today to be an immense gap.
What is the biologically relevant information content of the simplest living organism known to exist?
The information content of the simplest cell that we have today, I would estimate to be about 1 million bits. This would be typical of some of the mycoplasma type of organisms. I'm assuming that about for every base pair in the DNA of the genome of this simple cell, you have approximately 2 bits of information. Now, as to the estimate of what the minimal cell historically would have had to have possessed in order to be considered to be an actual living entity, I would put the figure down to no less than 10 to the 5th or 100,000 bits. I'd be willing to believe that you could put together a minimal cell with about a tenth as much genetic information in it as in the simplest cell today. I assume about 500 enzymes required for the minimal cell, about 100 amino acids for each of those enzymes, and then you just, a simple calculation gives you about 10 to the 5th bits of information, assuming no redundant DNA in the simplest organisms. And that's what we see today anyway. The simplest organisms use all of their DNA for genetic information.
How probable is it that such complexity could arise by undirected chemical processes?
Well, I think random chemical processes would have virtually no chance of generating the kind of specific order that you need in the earliest cell, in the biopolymers, in the proteins and in the nucleic acids. You just have to have a specific sequence of subunits. And I think you can show by some pretty convincing probability calculations, such as have been carried out by Hubert Yockey, that in the whole history of planet Earth, in 4.5 billion years, you would really not expect to find in the primitive oceans more than 1 or 2 or half a dozen at most functioning enzymes. This is not even taking into account the issue of having the minimum set of enzymes for metabolism, the 500, let's say, all at the same place at the same time. This is just a calculation at trying to see what the likelihood is of finding just one molecule of one functioning protein, or a half dozen at most. And so I think that, in my view, there's virtually no chance of chemical processes generating the kind of specified complexity that we need for the first living organism.
What is the most plausible scenario for a purely naturalistic origin of life?
I don't think there is one. I have reviewed many suggestions, and I can't say that I could really support or give credence to any of those that have been proposed. I think Miller's experiments do show us the possibility of getting some of the biomonomers we need, and that's to the good. I mean, that's a strong point of that methodology, Miller's simulation type of studies. But I don't have a candidate for the most plausible general scheme for going all the way to the first cells.
What are the major unsolved problems in origin of life research?
Well, I think one of the unsolved problems has to do with the optical isomer preference. How can we get L amino acids going into the first biopolymers when the experiments routinely show an absolutely equal production of the L form and the D form? The same situation exists with respect to the sugars in the DNA, and so I think that that is a most difficult and intractable problem. It has not yielded at all in the 4 decades that it's been studied. We know no more today about it than we did 40 years ago on that. Other unsolved problems involve how you could have the formation of the first nucleic acid molecules. We don't have a chemical pathway that's convincing to get either RNA or DNA formed on the primitive Earth. And then on top of that, we have not the slightest experimental indication of how these polymers, if they could have formed, could have acquired genetic information, this crucial problem of how you link the subunits together in just the order that they must be in order to code for proteins that can participate in metabolism. Not just any specific order in DNA will be biologically relevant order. It's got to be those sequences that have biologic meaning. So I would say that those are the two most difficult outstanding problems: origin of genetic information and origin of optical isomer preferences.
What is the relevance of the second law of thermodynamics to the origin of life?
Well, I think that the second law of thermodynamics does have some relevancy to the origin of life problem, but I do think that you have to be very careful in describing just the way in which it is relevant to the origin of life. The second law says that an isolated physical system, one that's not exchanging matter or energy with its outside surroundings, will, in every change that it undergoes, tend to go to a state of greater disorder or greater entropy, and at the same time the free energy or usable energy is declining. The chemical evolution hypothesis about origin of life on the surface of the primitive Earth, when you first look at it, seems to be sharply against the second law of thermodynamics, going from a relatively chaotic state of the primitive gases in the primitive atmosphere to these highly organized and complex molecular systems that are living cells. But you can point out, though, that the surface of the Earth is an open system, and it is receiving high-grade energy from the sun. So you could maintain that just so long as the decrease in entropy on the surface of the Earth is compensated for by an equal or greater increase in entropy in the total system, Earth plus sun, that it is conceivable that chemical evolution does not violate the second law of thermodynamics. The problem I have with that is that we have no empirical indication whatsoever as to how an energy capture system, i.e. a primitive photosynthesis mechanism, could have originated by purely natural means. And then, of course, we have no indication in the experimental data as to how a genetic system, a gene, which would direct chemistry against the tendency that the second law imposes on matter toward greater disorder. In the absence of those two lines of evidence, I'm going to suspend judgment about whether or not the origin of life violates the second law. But I think that just on the face of it, there is this huge problem. You know, the second law describes matter going to decreasing states of complexity, and the presumed origin of life by natural means is the reverse process.
Is it plausible that an RNA world was the precursor of the first living cells?
Well, the RNA world concept does have a number of attractive features. With the discovery that RNA has both catalytic activity and genetic information-bearing ability, you have a single molecule type that combines the key properties of the two great classes of macromolecule in life, the nucleic acids on the one hand, the proteins on the other hand. So many researchers have believed that the first living systems might have developed out of a situation where you just had the RNA polymers in the primitive oceans undergoing some kind of proto-natural selection toward the first cells. I think there are a number of problems with the RNA world hypothesis. One is, how do you get the RNA polymers in the first place? I don't see how given what we know about the chemical properties of the building blocks of the RNA, how they could survive in the energy rigors of the primitive surface. They would have been destroyed by ultraviolet radiation. But more importantly, how do you get these building blocks of RNA to link up in the natural manner to form the biological polymer in the face of potentially interfering cross reactions, which would have been much more likely in the primitive oceans? So I have problems with how you get the raw materials, the starting point for the RNA world. The second problem I have with the RNA world is that the catalytic activities that have been discovered for RNA are really highly specialized, few in number, and they have to do with complex events in the eukaryotic cell, processing that RNA in a highly evolved system. And I'm not sure that that kind of catalytic activity would have been relevant in a prebiotic setting where you'd need a vast range of different catalytic activities, sort of a generalized catalysis over the whole metabolic spectrum. So I do not think the RNA world concept helps us much in the origin of life problem.
Why must proteins in living organisms contain only left-handed amino acids?
Well, I don't have a completely satisfactory answer as to why present organisms need to have or must have just one optical isomer of the amino acids and the sugars in the DNA. I have some ideas about it. One would be that in order for enzymes to work and recognize their substrate, you have to have what's called a three-point landing on the surface of an enzyme molecule. And so if you had the D form of an amino acid interacting or colliding with the surface of the active site of an enzyme, the enzyme would not recognize the amino acid. If we had both D and L forms of amino acid in our polymers, in our proteins, and same thing for sugars in our nucleic acids, we would need, it seems to me, many, many more enzymes than we can get by with today in living systems, because you'd need an enzyme for the D form and then another enzyme for the L form of amino acid if both were used in biological processes. So it would seem to be very inefficient energetically. The cell would have to invest so much energy building all of these extra enzymes. So there is something to be said for the efficiency that you get when you just go down to one optical isomer. As to how this could have originated, we haven't the slightest idea. We don't find any asymmetric forces in nature which could have given us the isomer preferences before life started.
Why must nucleic acids contain D rather than L sugars? How is the problem of chemical chirality accounted for in naturalistic origin of life theories?
Well, I think that we have the need in current biochemistry for one of the two stereoisomers, the L versus the D, for the efficiency of enzyme action, and also to avoid having the need to have so many extra enzymes to react with the other isomer if both are present. So there's an energy savings there. But from a point of view of the naturalistic theories of origin of life, we haven't an explanation at all for how this current preference of optical isomers developed. Many experiments have been tried searching for clues as to how you might have gotten a preference of one over the other. And William Bonner at Stanford University, chemistry professor there, who has researched this problem for about 3 decades, wrote a review in 1991 of this subject, concluded it by saying we don't know any more now than we did when the search first began. It's just a very great enigma, a tremendous problem.
If life did not originate by chemical evolution on the primitive Earth, what other possible scientific explanations exist?
A number of suggestions have been made about other possible origin scenarios. They tend to go toward extraterrestrial origin, though. I think more and more scientists are recognizing the many, many problems with the chemical evolution theory for origin of life on Earth. And some of those are proposing that the origin didn't take place on the Earth at all, but took place somewhere else in the cosmos, and then the first living cells, microscopic microorganisms were transported somehow to Earth. We have the directed panspermia idea. I'm not totally convinced this was proposed totally seriously, but it's in the literature, and it's the idea that life originated on some other place in the cosmos, and then an intelligent life form shipped the first microbes to our planet deliberately, directed panspermia. So there's one possibility. Professor Bonner has suggested that the optical isomer sorting must have taken place in an extraterrestrial setting. He does not see how it would have been possible on the Earth, but we have the result of the sorting today. Chemical evolution must have happened somewhere, so it must have happened by an unknown means in deep space, and then the isomers transported down to the Earth.
What do you mean by your statement that perhaps scientism is more widespread than we like to think?
Well, I think that in view of the great many difficulties that we're now aware of in the theorizing about origin of life along the lines of chemical evolution, it is a rather surprising fact that so many scientists still insist that a chemical evolutionary origin will be found with future research. In my mind, it must go back to the general presuppositions that people make regarding this area. And the first presupposition is that chemical evolution must have happened, and we're going to keep searching until we find some kind of plausible actual chemical sequence. But that looks less and less likely now, and yet we still have a general agreement that it must have happened, and it's still uniformly taught that way in university science classes. And so maybe it has something to do with scientism, which I take to mean the belief that scientific method, as presently defined and conceived, can answer every question, including the great question of origins, origin of the universe, origin of life. And so that's what I mean by scientism being perhaps very deeply ingrained in our scientific culture, in our academic culture.
Is it possible that natural processes are insufficient to account for the origin of all biological information?
Not only possible, I think highly likely that natural means are insufficient to account for anything but the almost trivial increase in biologic information.
Can science rule out the possibility that most biological information had an intelligent cause?
No, I don't think so. I think quite the contrary, that it makes sense to actually reach the conclusion that from the data of origin of life studies, from the data of molecular biology, and from many fields in the of science, that an intelligent origin is a very likely possibility for the origin of the first life.
What alternatives are there to pursuing purely naturalistic explanations for the origin of life?
Well, I think it is conceivable, and it makes a lot of sense to think that the empirical data that we have in hand, taken in toto, all the different lines of evidence bearing on this problem of origin, to actually be in powerful support of the idea that the first life was actually created by an intelligent agent of some kind. I wouldn't be prepared to say at all from the scientific study who or what that intelligent agent is, but I think the data are in strong support of the possibility, or the likelihood, that the specific order of the bases in DNA, for example, the specified complexity, was actually impressed on matter by some intelligent agent.
What do you mean by intelligent design as it relates to the origin of life?
Well, intelligent design, I think we reason here by analogy, and I like to think of it along these lines that if one attempts to classify physical systems, complex and ordered physical systems, as to how they come into existence in the first instance and in subsequent instances of them, I think that you can find that the physical systems sort out into three categories. The first type of system, which I regard as the low information type of a system, would be an object in nature that could arise by purely natural means, you know, rock formations, snowflakes, crystals of all kinds, storm patterns. Many different examples exist, but these systems I call type 1 systems, and they can originate spontaneously by purely natural processes. Type 2 systems have to do with systems that humans are familiar with as manufactured artifacts. Everything from a paperclip to a supercomputer. There, the matter is arranged in such a way that we cannot see how it could have gotten that way spontaneously, because we can't deduce the order from the physics and chemistry of the components. Writing and language is also in this category. The pattern of letters in intelligible text does not come from the physics and chemistry of ink and paper, but it has to be impressed on the matter from the outside. So these I call type 2 systems. The first origin of an instance there is by manufacture, and the subsequent instances are also all by manufacture, because these are not self-propagating systems. There's a moderately high information content to these. I mean, the range goes up into pretty high numbers of information content. Now, the question is, what is a living cell? What does it fit into type 1 or type 2 order? I think it's a third category. The first instance, I think, has to have, of the first cells has to be by creation by an intelligence. But then once it appears, it is self-propagating, so it can, all subsequent instances come from the first one by purely natural processes. I think that in this third class, there may be one other instance. That's the von Neumann machine, the artificial device not yet manufactured, but a theoretical device which could be self-replicating, a manufactured device that could do its own mining operations, its own smelting operations, its own preparation of material, and then fabrication of itself, and then it could be a truly self-propagating artificial system. So I think that third class has two members, living cells and the von Neumann machine, possibly. Okay, this gives us a framework in which to answer the question about what we mean about intelligent design and the origin of life. It seems to me that the order we find in cells, especially in the DNA, and the genetic information, is more like type 2 order than type 1 order. And therefore, I would say the indications are very powerful that the first living cell must have been, well, for want of a better term, in quotes, manufactured somehow.
Why is an intelligent design or creationist interpretation of the scientific data not acceptable to many scientists?
Well, I think the reasons here, there'd be many reasons. We would go back to the climate of opinion at the time that Darwin's work was done. And remember, the battle there was between special creationists and those who wanted to have a completely, what they conceived of as a completely naturalistic explanation for this large area of science, namely the origin and development of life. And so Darwin's ideas were very appealing, I think, to many scientists who also wanted to further this program themselves, but couldn't see a way of doing it. And he came up with a naturalistic, a uniformitarian explanation that, given the information available in his day, seemed very convincing, and his method of argument was outstanding. He's a rhetorical genius, no question about it. Okay, so then you had a long period of time in which the faculties of the universities were Darwinist, and so deeply ingrained habits of thought in the succeeding decades, and so that now you find that that most of the professors, people working, research scientists, have been so deeply committed to these habits of thought that any suggestion that the naturalistic explanation may not be sufficient is to them like going back to the old special creation mindset that they thought they were gotten away from for good, with it finally accepting Darwin's view.
What criteria could be used to determine if the information content of living organisms had an intelligent or natural cause?
This is a current need in the research effort to try and define formal criteria, quantifiable if possible, that will allow one to decide whether a given type of order that's seen, or complexity we should say, seen in nature, is likely to have had an intelligent cause, or is rather, on the other hand, explainable by purely natural means. I think this concept of specified complexity is important, that you have to have particular order of subunits that have biological meaning, and if you find a system that has any order like that in it, then it's likely, in my mind, that it came from an intelligent source. Again, a reasoning by analogy to manufactured, the kind of order we find in manufactured items. Probably also we would bring in the probability would be quite low. Probability calculation for the chances of getting the same type of order by chemical means would have to be a very tiny number. Now, exactly how small it would have to be, I mean, that's something that has to be worked on. But we're at the beginning of that phase of things now.
Does academic freedom allow you to discuss the difficulties of scientific naturalism and origin of life theories?
Well, I think ideally, academic freedom concepts should allow one to freely examine the criticism of Darwinian evolution and the criticisms of chemical evolution in the classroom, and even to discuss the possible alternative interpretations of the data that lead one to an intelligent design paradigm. Unfortunately, I am not allowed to discuss these topics in my courses here at San Francisco State. It is not felt to be a legitimate part of a course outline or a course content to give counter arguments against the general Darwinian view, and certainly not thought to be permissible to give any kind of serious discussion of alternative interpretation. Now, this raises a very interesting question, and that is the question as to why this particular area of science should have this kind of immunity to critical examination. I would have thought that that we would have free inquiry in this area as we do in virtually every other area of university life, and for both big issues and small issues. We should have that same kind of give and take in the great debate on origins, because after all, the history of science is full of confrontation of major contending points of view. Just a couple of outstanding examples come to mind: Ptolemaic astronomy and Copernican astronomy, a tremendous debate at about the time of the switchover. Galenic physiology and Harveyan physiology, a tremendous debate in the early 17th century when that was, and you can name many examples. Those are quite large examples. There are others on hundreds of examples on smaller scales of the comparison that should go on all the time between major contending views. But somehow or other, in our current academic situation regarding the topic of origins and evolution, a wall has been built around the subject of neo-Darwinism, and so one is not really free to carry on a scientific critique of this topic.
How should the origin of life be taught in light of the California Science Framework Policy, which states that nothing in science or in any other field of knowledge shall be taught dogmatically?
Well, not dogmatically, obviously. Now the way it's taught now is very dogmatic in the sense that only one particular view out of several major contending views about origins is permitted generally speaking, in university classrooms and also in secondary education level classrooms. And if we were to follow the letter of the California guidelines now, then the debate would open up, and we would have this wonderful exchange of views, and we would have a chance to make more rapid progress in our in debate on this, and students would be better served. I think students need to know, not about every discussion and dispute that goes on in the scientific world. You can't expect them to be able to absorb that, but certainly the major fights, they need to have some information about those.
How is scientific progress impacted when critiques of current theories are suppressed?
Well, I think progress will be much less than it could be ideally. I think that in the climate of free and vigorous debate, you have the greatest chance of making the greatest strides forward. After all, there was a very vigorous debate at the time of Darwin's publication of Origin of Species. He won the day. He won it rather quickly, but there was a fight. Now we have a situation where we have the impression is given that the question of origins has long since been settled. And I deny that that's the case. I think that the more we learn in molecular biology and the more we learn from chemical evolution studies, the less likely is it that the chemical evolution explanation is going to turn out to be the right one. And so it's absolutely imperative now that we open up the discussion and have a serious debate on it.